All right. Hello, everybody. Welcome back to Archaeology with Flint Dibble. And I am here with a friend of the channel, Dr. Kim Foecke, an incoming assistant professor at George Mason University. And huge congratulations on that. And we're here to talk about the Homo naledi controversy. And I'm sure some of you know about that. We'll introduce it in a second. But first, we were just laughing a bit about her last name because it's spelled Foecke. And I was like, how do you pronounce it? Is it fuck? And she told me no, it's fakey. But her family, what happened? What's the history behind your name?
Well, it used to have an O with an umlaut, and then when we got to the US, everybody has trouble with the umlaut, of course. And so
Mm-hmm.
apparently somewhere along the line, they started mispronouncing it from Foecke or something like that to fuck, and that's terrible for English speakers. And so somewhere along the line they changed it. They decided to change it to fakey, and so we've been fakey ever since. And I think we're the only ones in the U.S. at the moment.
All right, so we have a totally unique pronunciation of fuck. Sorry,
Yes.
I mean fakey. So we're here to talk about this Homo naledi controversy. What about a year and a month or so, 2 months ago? Lee Berger and his team, they're excavating at Rising Star Cave. They published some preprints on some findings from these caves about Homo naledi, and in particular, one of them has generated a lot of critique and feedback. I mean, all of them have, but the one on burial in particular is one that where they presented a lot of data, but there's been a lot of issues with it. I might have been one of the first people actually to speak out publicly on it with a check out my video on my peer review of it, my public peer review, and looking at some of the issues of how they don't really prove effectively that this is a burial. So there's a huge controversy over whether, Homo naledi, there's all these preprints. It ends up on Netflix, whether it is actual burial of the dead, in which case this would be the first example ever, by far, of a hominin burying their dead, earlier than the examples from modern humans, earlier than contested ones from Neanderthals. And so they present a very complicated argument, but like some of this is just kind of simple logistics. The pit that this hominin is in is like 50 centimeters by 25 centimeters. How do you shove an individual in there, someone who's standing, more than twice that height? It sort of strikes me as problematic. The other issue, of course, that some of us have pointed out is the profile, the way they sort of publish this and they zoom in on it. There's this cut, but it doesn't look like a clear cut for many of us that study this kind of stuff. And then if you look at the earlier publications on the geology of this chamber, there's all kinds of evidence for sort of water movement and rolling down the hill and all this kind of stuff. So, they haven't really proven that it's not just slumped into a pit. And one of the biggest arguments that they came up with, which is where Dr. Foecke here has something to say on, is they argued that there's a difference in the composition of the sediment that's within the pit versus without. And so that makes sense. In a burial, you sort of shovel out some dirt and then you refill it, and the sediment's going to be different, perhaps chemically, perhaps in orientations. There's all kinds of ways that geoarchaeologists can study this. So, Kim, how did you get interested in approaching this? You're the lead author on this. So there's two other colleagues, but you guys are one of the only critiques that's actually gone in depth into critiquing some of the details. So, what inspired you and Alain and Robin to look at this data?
Well, I have to say that first of all, I want to underscore that the reason that I was able to pull this off is because I had the time. So I am not yet an assistant professor. I'm coming into assistant professor position. I've been in basically the equivalent of a postdoc position for a while now. And, I am not overwhelmed with the responsibilities that, mid-career and late career researchers have on their plates as far as university admin and teaching and all of that quite yet. And
Okay.
so one of the reasons that I pursued this was because I kind of felt like no one else would have the time and energy to devote the amount of effort that went into this to actually definitively sort of demonstrate what's going on here. But I first got interested in this because I noticed when the preprints came out that they used XRF or X-ray fluorescence to analyze the chemistry in this burial. And I'm actually acquainted with one of the peer reviewers who reviewed the original preprints for eLife. And she had reached out to me, and this person has not revealed their identity yet.
Okay, so it's not Jamie
So
Hodgkins, who I
it's
actually
not Jamie
interviewed in a
Hodgkins. No,
previous
it's someone
video.
else
Okay.
who has not revealed their identity, but knows me, knows that I published on XRF and archaeology, knows that I have a background that is not strictly archaeology. I came into archaeology from geosciences and material science. So I have a more sort of, how should we say, hardcore understanding of some of these underlying methods and the, physics, the high energy physics going on with these methods. And she was like, am I interpreting this correctly? And so before the preprints even came out, I saw some of the plots and some of the things in the preprint to try to help this reviewer understand what was going on and give a fair review of the paper. She didn't want to review without understanding and didn't want to misinterpret something. And so that's really when I got my first introduction to maybe how this was going to play out. I gave her my take. She put her take back in the peer review, and then radio silence, and suddenly we had CNN article, Homo naledi buries their dead and makes rock art. Meanwhile, the peer reviews are sitting with eLife for, I think it was almost a month.
Yeah.
Yeah,
More
and
maybe. Jamie was very upset about that when I talked to her because, like, this is one of the big issues behind the scenes. So, like, if you think about this behind the scenes, and I've talked to some people also who've not waived their anonymity, maybe even
yeah.
reviewers of a previous submission in different journals. One of the difficulties of peer reviewing in today's world is you get a paper that is extremely interdisciplinary. And what happens is you only have two peer reviewers. And so, but there's like 6 different disciplines that contribute. Maybe you have 3 peer reviewers, something like that. And to understand, like, I can only critique what I'm a specialist in, right? And so how can you critique other parts of the paper? And so, sort of behind the scenes, everybody was like, why is this being trumpeted in this way? There's been very clear criticism of these findings, and yet they're presenting it publicly as if it is a sure, proven thing. And it's sort of like, that's just kind of dishonest when they already have that feedback. This is one of the points I made in my first review is, this is not exactly an honest way to approach this. You have feedback here, and then you're claiming it's 100% proven. And the eLife reviews are now public and out. Who reviewed them is not, but the comments on it are public and they're out. And, they are scathing. They are very, very critical of the art of the burial, and at every level too. It's not just like one little thing, it's at a lot of different levels. And so the way this got so much press is just so shocking.
Yeah.
Did the press in some way inspire you as well?
Yeah, I think that I definitely wouldn't have been motivated to push this all the way to publication if there hadn't been so much press. And I saw, so many of our colleagues in the field were so frustrated about this. And you see, constantly we have people pushing out really amazing work in our field and really kind of sensational findings and amazing stuff. And, they don't get the opportunity to splash their work across the pages of CNN. And it's great work and it's good science. And the fact that we consistently see institutions like Netflix and National Geographic picking up individuals and research that are, not the most rigorous of the scientific representation that we have as a field frustrated me. And so I started to reach out to colleagues. Is anybody going to do something about this? And, as an early career person, I want to see, a good representation of our field going forward for my future career, for the career of my future students. Like it's about the integrity of our field. It's about the reputation of our field. And so I felt not only motivated to publish this paper, but I sort of felt an obligation to publish this paper because I have the expertise, I had the time, and what would be my excuse not to?
Yeah, and it's kind of strange actually to think about it because this is a critique of a paper that actually has not been finally published yet because the original peer reviews came out what last July, about a year ago
Yeah.
from now. And in theory, the team should be revising the publication, but if they have, it's not out yet a year later. So they've not been able to muster up a reply at all to all the critique that has come out. And there's multiple published critiques, lots of different critiques on social media or in editorials as well. It's kind of remarkable that there's so much peer reviewed now. There's yours is what the second or third peer reviewed response, I
Yeah.
think, maybe third
I
peer
think
reviewed
third, yeah.
response to this to a paper that has not actually we haven't even seen the final version, and that just is testament to the fact of how much press this got. It really they had a whole Netflix special designed around it, and it wasn't just CNN, it was all over the place. And I mean, I still get comments on my YouTube videos being like, oh my God, you're such a downer, doubting all this stuff. And it's like, it's not just me. The entire field kind of thinks that there's a problem with how these results were presented.
Yeah, and
And
that's the thing that kind of has also frustrated me is that when people come out with critiques, and I've even gotten some of it on this paper, I'm not questioning that there could be burial by a small-brained hominin species, right? I'm not criticizing the idea that this is possible. And I think that, it's important in science that we present ideas that might seem a little bit cuckoo to most people in the field at any given point in time, because, some of those ideas have gone on to become foundational staples in the field. And I think it's important to emphasize to the public that this is how science works. We get new evidence, we change our minds, we change our interpretations. Just because something was put forward 50 years ago doesn't mean that it's necessarily correct or has to stay accepted in the field. And it's just part of this iterative process of science. We're self-correcting constantly. And so I am not opposed at all to actual evidence that Homo naledi buried their dead or made rock art just because it had a small brain and it's not what we would expect in that place and in that time in human evolution. That would make me a bad scientist. And so what we're critiquing here is simply how the science was carried out and what the actual data tell us as far as how we can interpret and whether those findings are robust to how we interpret scientific hypothesis testing. So I think that's really important to make clear here. And I think that's something that the media often gloss over, like, oh, scientists change their minds. Scientists are, upended by this fact or that fact. And I think that rhetoric is really damaging to the public's understanding of how science works.
yeah, I agree. And I think also it's important to note that this isn't a personal beef in any way. Like,
Absolutely.
I mean, all of the scholars on this paper have done fantastic, phenomenal research on a wide range of topics, both with Homo naledi and the excavation of the cave and everything like that, and plenty of other sites and plenty of other topics. So it's not like it's
Yeah.
some sort of personal beef. I'm sure at times tensions run high among people that are being critiqued back and forth, but
Yeah.
it's certainly not personal in any sense. And in many ways, at least to the conclusion that I think of, is there's nothing wrong with a hypothesis that they buried their dead or the rock art or anything like that. It's a question of how definitively proven it is or not. Whether or not this paper ends up, I imagine some version of it will end up being finalized. It's just a question of how strong the conclusion is. Like, is it something that still needs future testing, or is it something that is proven? The original results made it sound as if it was proven. And as someone who studies bones, right? I study animal remains and I've studied lots of burials. I've excavated plenty of human burials in later periods that are very, very clearly burials that are monumental and constructed and stuff like that. They're under churches even. And so I've excavated and studied and analyzed this kind of stuff. One of the things that kind of shocks me is how only half of the burial was excavated. They're presenting a result from osteological analyses of skeletal remains, saying there's only one individual there with a few extra bones, but you only have a third of the bones from the pit actually excavated. None of them have been cleaned properly. And so to make that firm of a conclusion that there's only one individual there with a few random stray bones in there is like totally preliminary. That's something that needs documentation and proving. It's a hypothesis that could be tested with further excavation. And then they say, well, we don't want to damage it all, so that's why we left some of it unexcavated. But they have dozens of individuals across this cave system. You might want to excavate an entire burial, one of them, if you're trying to prove that it's a burial. And so, yeah, it's just a question of how strong these conclusions are. Unlike, say, Graham Hancock's Lost Civilization from the Pleistocene Ice Age, that I think there's clear evidence against it existing, as I've presented on Joe Rogan and elsewhere. But with this Homo naledi paper, I think that what it is it's a hypothesis that needs further testing rather than something that is definitively disproven. So let's jump into some of the details here. One of their key points is that the sediment in the pit, which they're claiming is a pit, is different than the sediment that's outside of the pit. And as we've already seen, the kind of features of this pit are not very clear anyway, but it's a hypothesis that it's a pit. And by the way, me reading this profile, I would not say it's definitively a pit, but I also would say that it's worth testing. So I'd probably excavate it and sample it different than the surrounding sediment to test that hypothesis. It totally makes sense to go and do that. That's how we do scientific archaeology. Here we go. So yeah, they carried out petrographic analysis of the sediment. And then they did bulk chemistry analysis using principal component analyses, right? And what this is they're looking at different components, elemental components, using XRF and XRD. That's what? X-ray fluorescence and X-ray diffraction, right? Yes. And so with that, you sort of zap it. And it gives you these spikes and these graphs that we'll look at some of those, and then they show the different elemental compositions that exist. And then they use a statistical type of analysis called principal component analysis to discriminate, in a sense, do these sediments in different areas look similar or different? Is my summary of that for the layperson accurate? Do you want to add or correct me?
This is one of the ones that initially kind of piqued my interest in this paper, mostly because I personally have never seen principal component analyses reported in this kind of display before.
Yeah, aren't they usually done as like dots that cluster? Am I wrong?
So
yeah, okay. I don't usually use it, but I've learned it in grad school. So yeah.
And so what struck me as interesting with this plot is that they've connected all of their samples together with this sort of line graph as though there's some sort of left to right relationship here between their samples, as if, you know, you went from the top of the section to the bottom of the section, for example, down through depth or something like that. That's how I initially, when I saw this, I was like, oh, okay, they're sampling through a section or through the profile to kind of get at the chemical differences between the different layers in this sediment in the cave. And then when I looked closer, the x-axis is sample number, and then the colors that you see here are simply delineating different areas of the cave. And so this manner of displaying this data being sort of non-standard and not something I've ever seen, and maybe one could argue a not particularly advisable way to plot this type of data for interpretation. This is kind of what tipped me off that there was something worth investigating further in this paper. So this figure in particular was kind of the jump point for me to dig into the supplemental information and dig into the raw data and try to kind of, I started by seeing if I could just replicate, right? The cornerstone of science is replication. And when we publish a paper and put the data out there for the scientific community to see, the goal typically for most scientists is to provide enough information in that publication that one could redo the experiment to some degree and achieve the same numerical or statistical or whatever output. Now, whether you interpret that output the same way as everybody else is a matter of debate, and obviously different scientists have different ways of interpreting data, but we should at least be able to get to the same numbers and the same statistical result that the authors of a paper put out. And so we started with that. Can we actually replicate what they have generated here in terms of plots, in terms of numbers, in terms of statistical separation between the chemical compositions of these different sediments?
And so I just want to clarify really quickly that this is the data that you're trying to replicate in a sense, right? So this is the table.
So this is the raw data. This is what we're using to try to replicate.
Yeah.
So those other types of plots, like the one you showed previously, where they've got, their samples and their principal components on the y-axis, or there's some bar plots in the supplemental that show the different loadings on the elements, how much they contribute to the
So those sample numbers correspond to this column. And then the different elemental percentages, is that what we're reading here?
So these are weight percentages of elemental oxides. So basically this means that the number percent of the sample, weight percent, is that elemental oxide. And so this is the raw data. We're not trying to replicate this data. We can't replicate this data. We don't have the samples in front of us. I can't re-zap this in my lab.
And chances are that this is probably okay, because they're literally just, they're taking a sample, they're zapping it, they're reporting the results, and then it's okay. Well, we'll get into that in a second. One would hope that this should be mostly okay, but yeah, we'll talk about that in a second.
Yeah, we can talk about that. That kind of gets to this whole, one of the things that you mentioned is like the peer reviewers had all kinds of issues with these papers, and the issues are kind of at all levels, right? So they've got very high level issues and they've got very nitpicky issues. But inside of this analysis, this is, we're talking about like a third of the data presented in one of 3 peer-reviewed preprints. We still have issues, I think, at all levels. We have issues from research design to data acquisition to method choice, and then down to analysis of data, visualization of data, and interpretation of data. So at every nested level of research, from the highest level where you design the experiment and choose the methods, through actually collecting the data in the lab, all the way to how you visualize it, how you analyze it, and how you interpret it. All of those, we flagged issues in our paper for just the X-ray fluorescence data.
Which is a tiny segment of the original paper, yeah. So let's look at your guys' replication attempt. So this is what was published, right? And we looked at the table that you then took those values and you tried to kind of replicate this. And this is kind of how you would normally, I imagine, do this kind of loading of the principal component analysis, right?
So somewhat. So this is actually a different kind of plot. So on the left here in the bright blue column is actually the figures published in the supplemental of the preprints in the Berger paper, just as they are. We screenshotted them and we stuck them.
Oh, that's from the Berger paper. Aha. Yes. Okay. I see what you're saying.
Yeah, it's a little bit complicated to watch.
Yeah, well, exactly. That's why I wanted to make sure. So then this is an actual principal component analysis. Correct. And this is what an actual principal component analysis normally looks like when you put it through like R or whatever statistics software you're using, SPSS, and you create these different clusters of related and not related groupings that the statistical analysis creates those groupings for you based on the values and some of the presets that you use. So what's going on here then? This is you now looking at this. How is this similar and different to what we've seen the authors present?
Yeah, so when the authors presented that sort of line plot with all of the jaggedy peaks and the stuff connected with the lines, you could kind of see they were presenting the data in such a way that there appeared to be visible differences, right, between at least the different parts of the cave. in
Hmm.
terms of chemical composition. So you can see here that most of the brown squiggly line in the yellow and the green areas is above the 0 mark, and then most of it is below the 0 mark in the red and the purple segments here. And so the way that this is presented is giving you the impression that there's some discernible differences, right, in chemical composition between these different parts of the cave. And then when you kind of look at the yellow plot here, so the yellow plot is what we actually care about, because
Hmm.
they're not necessarily, at least to my understanding, at this juncture, they're trying to demonstrate a burial by demonstrating differences in sediment composition through the layers of sediment right where that skeleton is, which is our feature one, right, in the site. They're not trying to demonstrate that the sediment in feature one where the skeleton is different than in the Lassetti chamber, like down the hall in the cave.
Far
And
away, yeah.
so what we actually care about is this little section in the yellow on this
Hmm.
figure. And so what I did first was to strip away everything from the analysis that wasn't directly related to that skeleton, because it's going to create more variation in the statistical analysis to include, of course, different areas of the cave that are like hundreds of meters away from your burial, but it's not actually going to tell you whether you have any variation in the depth of that burial area itself. And
That
so,
makes a lot of sense. And I agree because the way principal component analysis works is it discriminates into these groups. And well, if you have a sediment from hundreds of meters away in a very different chamber, that's going to throw off the variation within your samples, which is going to delineate differences differently because
yeah.
that's how it works. And so you really need to just be comparing what's in the grave and what's right near the grave to see if it's different. You can't add in extra variation. You don't want to include sediment from Montana because
Yeah.
that's not going to be very useful. It doesn't
Yeah.
help you differentiate between what's in the potential grave and what's right next to it.
So this
Yeah.
was kind of one of our critiques of the way the research question was structured, right? Because this goes all the way back to like, how are you structuring your hypothesis? What kind of data are you collecting? And what do you expect to be able to ask of that data? And so if your research question for these elemental analyses is based on sort of a not quite cohesive understanding of what's going on in the site and what's going on with these methods, you're going to end up with something wonky. So we did actually in these plots include all of it, right? So
Okay.
this was just our straight up replication. We included everything the same as they did, but you notice in these plots, the different colors roughly map onto the different colors that were in that jaggedy line plot for different areas of the cave. There's much less apparent separation between the colored areas on this plot and the colored areas on their plot. There's a lot more overlap
Yeah.
here in the chemical composition of the sediment samples from not just the area right around the burial feature, but like the cave at large. There's a lot of overlap in chemical composition in this type of analysis. And so
In
when
fact, it
we
looks to me, I don't know what I'm doing with this, but it looks to me like the stuff from the very different chamber, Lassetti, is the stuff that's clearly differentiated,
right.
while the material from the same chamber, it looks to be fairly clustered.
Yeah. And so the more you zoom in on this plot, the kind of smaller area you end up having sampled. So we've got our you know, SB samples, or the green samples there right in the middle, and then the SC and the DF is above feature one and feature two. And everything is just kind of clustered right in that area where the purple is. So the yellow completely overlaps the purple, almost completely overlaps the bright blue, and those are all the areas that are right in that feature one area. And so we're not really seeing any structured variation at all. between any of the samples taken through the stratigraphy. So the samples that are at the top of the burial above the body are looking the same as the ones right next to it, are looking the same as the ones below it, and are looking the same as the sediment laterally right outside the body.
To the south, yeah.
And so, this partially, I think, comes down to, again, research design. There were very few samples actually taken from the sediment inside and around feature one. So if one were to do this in a more robust manner, you would want to make sure that you have a decent number of samples in each layer that you could identify visually. You would want to make sure that you sample laterally, so outwards on the ground level plane, all the way around the burial. Make sure that you've got continuous stratigraphy all the way around so that you could even detect a burial. So this is another thing that we've got going on. So yeah, so once you strip away all of this extra variation and zoom in on the variation right around feature one, we see, not very many samples through the actual stratigraphy. So
Yeah.
SE is the bright blue dots here are the like 5 samples that they took marching down through the depth of the stratigraphy to try to demonstrate that there's, differentiation here. And so in order to even ask a question of if there has been a pit dug through stratigraphy, you have to have stratigraphy, right?
Yeah.
You have to have these layers, like a layer cake, that are distinct enough that you can tell the difference between them. Because if you can't, you're just sort of, you could have dug through, dug a pit and put the sediment back in. And if it's all the same chemically, it's all going to look the same out in the wash. You won't even be able to detect a burial, even if there was one.
Which
And
is the
so,
point that this doesn't prove burial or disprove burial.
correct.
It just shows that the sediment is similar, which would make sense if you dug a pit and refilled it with the same sediment. However, what it does is it shows that there is no sedimentological difference between what's in the pit and with outside of the pit. Yeah.
Right, which is the cornerstone for the conclusion here, that they think they see a difference, and we think that that difference is not there when you analyze the data according to standard field methods. And I also want to make it clear that this analysis that we did to try to sort of improve on and do this correctly is not something we, kind of just came up with out of thin air. This is something that is quite well established in the field, and how you would go about analyzing data like this, how you would go about structuring research like this. So we're not, critiquing post hoc based on something that isn't published and that we're just now presenting. We're implementing a protocol that is how we teach people to do this type of work.
So in a sense, you're taking what they did and you're trying to say to everybody else, okay, let's think through a standardized way to actually demonstrate this so that we're all on the same page and we have agreed upon methods that seem to work and that can help demonstrate, all right, this sediment's different from that sediment. That would prove it's burial or not, right? Or at least prove that they're different. So that's really important. So in a sense, I guess about what, a third of the paper that you guys published is kind of like, all right, let's look at the data from the Berger et al. Burial paper in detail, in depth, let's say, and then let's see what it shows. And it does seem to show that there's no sedimentological difference, which of course is the title of the paper. But to make this more valuable, because people are, obviously if you're listening in, you're probably interested more in Homo naledi than in just how do you do geoarchaeology? But if we want to be able to look at questions like Homo naledi or other questions related to burial or sediments or whatever, if we want to take a geosciences approach to the archaeological record, we need to develop best practices and methods that we can all use and be consistent and compare and replicate data and stuff like that. Do you have anything else you want to say about the Homo naledi study before we move on to some of your recommendations for everyone else?
Yeah, I mean, there's a lot of, there's a lot more detail to this paper that, we we dug down into the actual data acquisition and how, the actual lab methods function and what was or was not done that may actually call into question the raw data that they published. And,
You mentioned that,
that's
yeah.
probably a little bit over the head of those of you guys who are watching this video, but suffice it to say that, the research design part of this is only one component of the critique. We also zoomed way in at the expert level and broke down how these methods function and how we think that the samples were analyzed in the lab and what we think may have been missteps in the lab itself that could have resulted in those numbers in the table that we used to redo the analysis. Those numbers might not even be an accurate representation of the elemental composition of the sediment. And
Okay, well, even just for my sake, because like I work with
yeah,
people that do XRF and XRD.
yeah.
So I mean, I have a paper where I work with a geoarchaeologist who's done XRF on soil to demonstrate diagenesis, how bones have been impacted by the burial environment in a sense, and the remodeling of the soil and dissolution and all this fun kind of stuff, right? So I have some understanding of this, but the few times I've seen people do XRF or XRD, they basically zap it. You get this kind of plot. You first assess it to see if there's any kind of issues, and you strip away any sort of the outliers or whatever, but then you end up producing this kind of data. But how would this data actually be wrong? Why should we not trust it when sometimes this kind of raw data is presented?
Yeah, so the main reason that actually what we conclude in our paper is that we aren't sure whether or not we should trust it because the authors haven't fully reported the methodological parameters that they use to collect the data.
Okay.
So in this type of methodology, a lot of the archaeological community views XRF as like point and zap, get a number back, and that number it means something. But there's a lot of considerations that you have to take with the point and zap instrumentation. So X-rays are funny little beasts. X-rays move through materials in ways that can be a little bit unpredictable. And X-rays, when you're calculating the percentages or the concentrations from your raw signal that's just X-rays coming back at you, there are corrections you have to apply for things like the matrix. So what kind of material is it traveling through? Is it traveling through loose sediment? Is it traveling through packed sediment? Is it traveling through a sample that you've completely melted down into a glass to analyze it? Is it bone? What is it? And all of these different materials interact with X-rays differently. They absorb some of the X-ray spectrum that would give us elemental information. They can block it from getting out of the material and reaching the detector. They can scatter it in different ways so they can shoot off to the side. They can bounce off some atom in that material and bounce to the side, and it never enters the detector, so you never actually collect the data. And the way that we deal with this in the lab is to use standard reference materials. So standard reference materials are a cornerstone of analyzing things for chemistry, for elements.
Yeah, use them for isotopes. Yeah.
Yes, for isotopes, for anything. And so these are materials that have a composition that for sure, 100%. And most of the time, these materials are things that are manufactured specifically with a certain composition to be standard reference materials. And they help to kind of create some predictability into the unpredictability of how X-rays are interacting with the material. So you'll want a standard reference material that is somewhat similar in structure to the sample that you're analyzing. And so if you're analyzing sediment, you're going to want a sort of sedimentary or rock-like standard reference material that has some of the same elements in it.
So did they not report what their standards were?
They didn't report what their standards were. They didn't even report if they used standard reference materials. So one of the problems with XRF becoming so common in archaeology is that we've also gotten common in archaeology some shortcuts that have been presented by the manufacturers of these instruments to make it easier for non-expert users to get data from samples without understanding all this stuff about X-ray attenuation and choosing standard reference materials. So they'll include in some software some sort of general or standardless calibrations where they'll take sort of a very high level guess essentially at a correction for a particular kind of material, and that gets applied to the data. And then what comes out the other side is a number, but that number may or may not be truly an accurate reflection of the chemical composition. And so one of the things that I learned when I was learning how to use XRF at a national lab with material scientists is never to trust those automatic, or calibrated factory settings. But we never learn that in archaeology. We see, a button in the software that says, calibrate my data to sediment, calibrate my data to obsidian. We'll press the button. It applies this kind of random calibration that isn't specific to our analysis, and it can have wild effects on the final data values. And so this is kind of one of our main points for the scientific community to take away from this paper. We've got some, public-facing conclusions about the, Homo naledi behavior, but we've also got some conclusions here for the scientific community. And one of those is A, report everything per, per field standard. And B, if you're going to incorporate a method into your research as an archaeologist, it's your responsibility to make sure that you learn enough about that method that you can design your research question properly, and you know what the data that you get back actually reflects. And so a lot of people deal with this by working closely with subject matter experts. So they'll work with a XRF specialist who's a material scientist or who's a geologist, and not just send the samples out for analysis and then get the numbers back and try to interpret them on their own. And we don't know what was done here. We don't know why the decisions were made. We do know where the samples were analyzed. They were analyzed in a geoscience lab. But, that's about it. We don't know anything else about how the data were collected. And so our conclusion basically is maybe these numbers reflect the actual composition of the sediment, but just as likely, maybe they don't.
Yeah, no, that makes a lot of sense. And I mean, it's a big problem in my field as well, where you get PhD students that are very excited, and PhD students are fantastic, but they get very excited to try using a new method, like, because you can use XRF to understand the composition of different metal alloys, very common in sort of the classical Greek world. And so you get students who really don't have any training in geosciences, and they just start pointing XRF guns at a variety of artifacts. But you need to understand the real basics of how this works. And so that's one of the key things. If you want to do archaeology at a high level, you need to really dig into the depths of how something works. I'm learning this the hard way, as I've realized that the way we do analysis of oxygen and carbon from tooth enamel with isotopes, it's actually really problematic. And my entire research design has changed for my project. And instead, I'm working on developing new standardized methods for how we can do this so that we have results that are replicable, because we're learning that they're not. And so it's a big problem for lots of people. If you do isotope analysis from tooth enamel, beware, there's more coming in the future. But
I'll be looking out for that, because that's actually my main research area.
Okay, well, we can talk about that in a different video once I work out more of it. Yeah, so there's some issues, and you really need to get in and understand, in my case, how powderized enamel might interact with the atmosphere, for example. These kind of basic things, which you don't always necessarily think about, because everybody just grinds up enamel and puts it in a tube and then throws it in a mass spectrometer. And so
I want to acknowledge, it is hard, as we are becoming such an interdisciplinary field. We're using 5, 6, 7 different analytical methods in one research project. It's so hard for a head PI to stay, informed to the level that you would want to be on all of these different methods. And it just really, not trying to particularly rag on this team for it. It's a widespread problem, but it's hard. It's hard to keep up to date. It's hard to make sure you know enough. It's hard to, get the right people involved in your project. And, it's something that we have to continue to sort of grapple with as we continue to become method borrowers, borrowing methods from physics and chemistry, material science, geology. Like suddenly you have to have 6 PhDs to do one archaeological site.
Well,
So
no,
not,
it's true. I've
yeah,
always told students and the public that, look, archaeology is by far the most interdisciplinary field. Any field of study out there contributes. It could be studies of literature and art on the humanities side, or it could be studies of organic chemistry and geosciences and biology and astronomy on the physics and everything like that on the hard sciences side, and all the sort of social sciences in the middle, the kind of economics and statistics and math and computer knowledge and software programming and statistics that you need to be able to know. And so, we really do borrow from every single field, and therefore we all have our own strengths and understanding, and it's why we need to really reach out. So that's a question I have for you. Did
yeah.
you or your co-authors ever reach out to the Berger team to ask them about how they did some of the stuff? Or have they replied to you now that your article's been published? How is that going
Yeah,
on?
so we chose not to reach out to them in the process of writing the paper, mostly because we really wanted to underscore the replicability aspect in what was already published. We shouldn't have to reach out to a scientific team just to obtain enough information to replicate their results. And that was part of the point that we wanted to make here. We didn't want to include information that we may have gotten through another channel other than the publication. We wanted to show here's what happens when you only use the information that's presented to the scientific community. That was a choice we made. Some people have given us a little bit of flack for that decision and said we should have reached out, we should have, asked questions. There's some merit to that, but that's just not the route that we took for this particular paper. I have heard nothing in terms of a formal response from anyone on the team. I did an interview with a news outlet the other day, and that reporter has since informed our team that they have gotten a response from one of the authors on the Berger preprints with, I think the quote was, a long list of objections to our paper. So it will be very interesting to see if or when we actually get any of that dialogue going. And from this point forward, I'm certainly not opposed to opening a dialogue with the team if they want to, have a chat with us and like talk about, how this research might go forward and how you might design research with these methods to ask the question that we are all wondering: do we have deliberate burial here? Certainly, like you said, this is not a personal beef. I've met Lee Berger. He's a nice guy. So I mean, I would be willing if he wanted to, have a chat about that. I'm certainly not going to be argumentative or combative about this situation. We're simply critiquing the science, and the hope is to make science better. And it's a really cool site, and there's a lot of really cool things you could ask of said site. I'm hopeful that there's a way forward where this site can give us some really amazing information and discoveries.
I agree. I think it's a really cool site. It's overall a good project, and I think all their hearts are in the right place. And I think that in many ways, this shows the value of peer review in a sense, because, I mean, I've gone through peer review, you've gone through peer review, we've all gone through peer review. The real goal there is not to judge it yes or no. It's typically to critique it and improve it, and to say, hey, this stuff is not clear. It's not, sometimes in a peer review, you might not usually go quite as far as you guys did in your review paper, but
Yeah.
I've seen people replot my stuff and ask questions or reanalyze stuff. That happens all the time. Hey, clarify this. What was your standard? That's a simple kind of thing that usually comes out in a peer review. Hey, this sort of basic information, which maybe you thought was extraneous and wasn't needed, well, it would be useful. So could you please share that in the final version? And so, that's usually how peer review works. I mean, I don't think I've ever given, as a peer reviewer, ever given a recommendation of rejection. At the worst, I've always done major revisions as my recommendation. I have then had the editor afterwards reject a paper, but only once, I think, as far as I know.
Yeah.
There's one paper that we were talking about beforehand where I never heard what happened to it.
I do think that, like,
Yeah.
we should acknowledge that peer review does have its problems, and that is one of the things that, the Berger team and others in the field have kind of centered in on. However the motivations for going open access come about, there are arguments that peer review holds science back or isn't, doing anything for the quality of science, because you can get bad reviewers, you can get non-expert reviewers. The system is quite broken in a lot of ways. However, I think that, overall, the overwhelming majority, at least in my experience, like, peer review has made my work better. There are absolutely instances where peer reviewers have not understood something that I've done, have not been enough of an expert in the subfield to actually review the work properly. And that's a problem too. What are the odds that out of 3 peer reviewers, you're going to get people who have the breadth of experience to review a paper like this in enough detail in all the facets? If I had gotten this as a peer reviewer, I would have a hard time having reviewed the skeletal aspect of this. But I could have done the XRF aspect to the detail I did here. But maybe a lot of other archaeologists would have glossed over the XRF and assumed, this is fine, and not really been able to break it down in the way that we did. And so I think that's part of the problem with peer review, is that as a field that is so interdisciplinary, we have to work harder to make sure that the peer reviewers that are selected to review papers actually can review all parts of the paper. And I don't know what the solution to that is. And some people say, well, the solution is preprints and open access. And other people say the solution is more peer reviewers per paper. And some people say the solution is burn it all down and start over. I don't know. But so I think that it is worth acknowledging that aspect.
Yeah, no, I agree. And I don't think peer review is perfect either. There's all kinds of issues with it.
Yeah.
And all kinds of weird issues with it. I don't think there is any real good solution. And I think in general, from where I'm sitting, and I think where you're sitting and where some of the other people are sitting, is the issue here might not even be the open data presentation of stuff. It's how it was presented to the public as kind of definitive. I think that is a different kind of issue. It's how do we present something? And I mean, we're all kind of guilty of it to some degree. I certainly, when I present a conference paper before something has been published in depth, I'm presenting an argument, and I'm presenting it in a clear-cut fashion, as if because I believe that that's what the data shows. And that's what Berger and other people on the team's response was that it's like a conference paper, right? And that makes sense to a certain degree, except that they'd already had all this kind of critique that they knew about, but the public didn't know about. And they'd done a whole sort of campaign around it that led to so much publicity. And I sort of think about preprints in COVID, where the media took some preprints and ran with it as if it was settled science, and then that made the scientific community look bad because it shifted what they say, and and it's confusing messaging for the public. And that's what this resulted in. It is a lot of confusing messaging for the public. And in many ways, I don't think that's really I don't know what would have happened. I can't be sure what would have happened if, let's say, the preprint came out with the eLife thing, and then ideally the reviews should have come out at the same time. Then the news would not have been as big. But instead, it came out as just the preprint and one side of the story and a press release. And the press release designed to gin up excitement for a Netflix special. And look, I want people interested in my field. I love it when people are interested in archaeology, but I want them to be interested in good archaeology and how we do it right, and about an honest approach to how we how we actually tackle this in the scientific community so that they can be informed in what we do and become better understanding of what we do and why we do it and how we do it and why it matters and why it's relevant. So yeah, I I guess
Yeah,
I'm running
and
out of
I mean,
steam.
it's interesting because in, doing some press for this paper, I've talked to some of the journalists who initially published the sensational pieces in response to their press releases, and the journalists are upset for a whole different reason. And the journalists are upset because they didn't have access to the peer reviews at the time they were doing research for their article. And as part of journalistic research, they would have come across those peer reviews, they would have had access to them right in front of them, and they would have made different decisions about how they presented their articles, how they wrote, headlines, how they spun the story in the media. And so I actually have heard quite a bit of discontent from some of these journalists who feel almost as if they were misled. And I think that a lot of things came together here to create a not so great situation, one of which was, as you say, this huge gap in time between the press release and the release of the peer reviews. And again, from what I understand, there was no real reason for that gap to occur.
That's what
Um,
Jamie said on this channel. Jamie Hodgkins, who was a peer reviewer, she said that all the reviews were in before the initial press release. So everybody within, and I didn't know anything about it at that time. But you did, I guess, because you
I
were
did.
you were in contact with one of them. So everybody with inside information about this process, from the editors to the authors to the peer reviewers themselves, understood how complex and nuanced these results were, and yet the journal and the authors went ahead with the kind of way that they did it. It's just so strange. I look, I respect them
Exactly.
for a lot of different things, and everybody makes a few mistakes, and I
Exactly.
get it. I get it. They were hoping to gin up excitement for the field and for their discovery, and I totally understand and respect that part. But let's think about the press response, because beforehand we were talking about how, the initial results got so much press. And since then, there's been this is the third peer reviewed article to come out critiquing those responses. There's been all kinds of non peer reviewed responses from real scholars who have, your co author, Alan Kefelec, wrote a nice Twitter thread about it. Robin Pickering has written a series of op eds and I think a peer reviewed article as well, a different one that she was a co author on about it.
Yep.
And so, a whole lot of us have sort of alerted the public, hey, these findings are problematic. They're not disproven, but they're problematic. They're not proven, let's say. And yet there's been very little press about this retraction. Why do you think the press is so focused on the initial news and then doesn't like to cover sort of like the blowback, let's say, which I find odd, right?
Yeah. I mean, man, because it's not cool and it doesn't have Disney money. It's cool to say there's burial by a human ancestor who's not us at this incredibly ancient time in this really, enigmatic, weird, cool archaeological site. It is not cool to go and say this scientist published a paper with really boring graphs in it that kind of walked back this idea. So I get it, kind of, from a journalistic perspective. Like, if I were to try to write a really splashy, exciting, news piece about this paper I just wrote, I would have a hard time doing that. So, of course, it's part of the nature of the media. They want the most exciting, the coolest thing that's going to get clicks, and part of what this paper does that I think has resulted in some journalistic interest is that it stokes controversy, which the media loves. But beyond that, I think there seems to be relatively little interest from the media in doing harder work to correct a narrative than, putting one out there in the first place. And it's frustrating to some degree, but also understandable to some degree. And you would hope that the subsequent work on these findings would get just as much of a splash. But at the same time, I don't have the connections. I don't have the platform. I don't have the sort of stage presence and charismatic pizzazz that some of, the authors on these other papers do. And so at the end of the day, to a reporter, I'm just not that interesting, which is unfortunate.
Nah, I think you are. I think that we all undersell ourselves as someone who tries to go out there with a little bit of pizzazz and charisma. I actually think it's very similar to teaching an intro-level archaeology course. We want to go out there, and it's even similar to writing a damn grant proposal. As my dad always taught me, when you write a grant proposal, pretend you're NASA trying to convince Congress to invest in a new telescope. Right? You start off by saying, we want to discover the wonders of the universe. You don't start off with saying, well, telescope technology has improved a lot in the last 10 years. You start off with the wonders of the universe, and then you work on down. And it's the same thing with your first lecture in Anthro 101 or whatever you're teaching, Introductory to Archaeology. You start off with that kind of big picture question, and then you move into something more precise. And so, in many ways, I should have started off this damn video differently, saying this is all about human evolution and the behavior of hominins, and it's going to help us understand that kind of stuff. I just assume, since Homo naledi has nice buzzword to it, people are already somewhat familiar, and so in that case, my intro might have worked. But because you're not going to click on this, Homo naledi's right there, that's why you click. In that sense, I think that relates to another bigger issue as well. Sometimes we have this sort of rut in what people know about and what journalists know about and what YouTube algorithms know about. And Homo naledi is one of those things, which it's great. It's a phenomenal site. It's a phenomenal new discovery, but there's also all kinds of other really phenomenal discoveries, stuff that you're working on, stuff that our colleagues are working on, that really don't get the attention that really should also get equivalent attention. And so we need to get better at being those kind of show people to get journalists interested, right, in what we
100%,
do.
yeah.
So
and nobody teaches you how to do that.
No, you have to learn it yourself. And so if any journalists are listening, what's the one most exciting thing you're researching?
Who, me?
Yeah, you.
The fact that maybe Neanderthals didn't eat mammoth as much as they thought, as much as we think that they did.
Okay, that's pretty cool. So the Neanderthals were not eating much mammoth. All right. I never would have. Yeah. All right. I'm cool. Are you using what? Isotope analysis to look at that? And
I am.
faunal
and
analysis,
I'm using the idea that Neanderthals may have cooked a large proportion of the meat in their diet. And when you cook meat, it changes the ascribed proportion of different prey resources in the diet in our reconstruction models and bumps mammoth way down the list.
Interesting. All right. So Neanderthals were not into hunting such big animals as we think.
As much
as we think. All right, cool. That is Dr. Kim Foecke. Thank you so much for this intro into the sedimentological problems with the burial of Homo naledi. Cool.
Yeah.